So the topic is digitizing ourselves, and what do I mean by that? I guess some of you or many of you here have some kind of device where you keep track of your sleeping behavior, your heartbeats, maybe insulin levels, these kind of things. But that's not what I'm going to talk about. So I want to take it further. And imagine that you can start to get genetic information out of yourself instead. And even molecular or proteomic information like this guy here before talked about. So at Stro Technologies, we are basically developing a sequencing machine for the future. And well, my name is Johan Strömqvist. I'm the CEO of Stro Technologies, and I'm here to describe a little bit about why, a little bit about our company and the background, and also why I think it's important with this digitizing ourselves to get genetic information out of us. So I'm studied here at KTH in engineering physics. Then I did a PhD in the border between medicine and physics, kind of interdisciplinary. So I'm not so fond of the term life science, but that's another story. So I was working with a kind of imaging technology. More specifically, it's ultra-sensitive imaging, which probably don't say you too much, but it's about image when you have very, very low noise level in your camera or in your sensor that you're looking at. So, and I was interested in looking at very, very dim objects, sort of, that doesn't give so high signals. So it's very difficult to distinguish if there is in your image, let's say a dim object like a molecule or something like that, and compared with a noise level. So out of this image, you probably won't get anything. But if you have very low noise level and you just keep repeating, taking images over and over and over again, you can start to generate a lot of data, and you can use computers to determine like information such as how many molecules you have, how are they moving around, are they interacting, this kind of information. So you can't see it, but bringing the data to computers, then you can actually extract a lot of information. That's the key concept, which I think is I will continue to talk about here during this talk. So what I was particularly interested was to apply this tech or this imaging technology on studying live cells and see how different drugs, how they affected signaling pathways within cells. There's an organization called STING, Stockholm Innovation and Growth, a kind of accelerator, which I can recommend if you want to build a startup here in Stockholm, a tech startup. So I went to that program 2010, and the business idea was basically to test drugs from pharmaceutical companies on cells from patients. So not directly on the patient, take out the cell. No, that sounds horrible, but using cells from patients and trying out drugs on the cells, and then and do that before entering clinical trials, before testing on humans or even animals, and to see what's actually happening. And so that was the business idea. The only problem with this kind of idea was that this imaging technology is really cool, but it's extremely slow. So it was hard to build a company out of that. So in, I think it was 2013, 2014, me and my colleague Annika here, we tried to rethink how can we make this faster, and we met this amazing guy, Bengt Sahlgren, who is, well, a very smart person. The professor at KTH loved him in laser physics. So, so we start to rethink imaging, and we actually, a bit modest here, broke the eternal triangle of imaging. And what does that mean? Well, the eternal triangle of imaging is basically, it's speed, resolution, and sensitivity. It's very hard to combine those three. What do I mean? So if you have a camera, using your iPhone or whatever, and you take a large image, it's very, very hard to take a large image and, hypothetically, let's say that you take an image of Earth and then zoom down to individual person. Very, very difficult to do that. Well, that's Google Earth. But it's a tedious process, a lot of stitching and years of blah, blah, blah to develop that. So it's hard to combine resolution and speed. But also sensitivity, that is a little bit more difficult. But if you imagine that you take an image, a photo with dim objects or in dark kind of darkness, you need longer exposure times. And that's means that it takes a long time to take the image. So it's hard to combine resolution, speed, and sensitivity. And that's what we did. So we kind of solved that. And that means that we can take like biological samples, DNA, cells, whatever, and transform it into a digital format which computers can read. And this is how it looks like. I can recommend you to go into tinyurl.com at ydepcube with K into U, and then you will see how it really looks like if you want to be impressed. I think. So I just took a few images there. This is a piece of brain that we got from our collaborator group, Professor Mats Nilsson. It's a section. Here, we take an image of this with our system, and here we zoomed in. You can see it much better if you go into the URL. But what you see here, so this is actually one photo that I saw. This is the brain, but you can zoom down to not only cells, but also genes. So we can see which genes are on and off within the brain at the moment it died. Sorry, the mouse. But we can also look at cells from blood and so on. So you can get genetic information out of the image. And this is a concept. So I tried to make a shift from thinking of images as just beautiful stuff that are aesthetic and so on, to more it's data. It's pure data. And it's intended for computers to read this data. And that was a professor here at KTH, Oslund. He was working a lot with imaging, real pioneer in imaging. Also founded a company called Sarastro, one of the first confocal microscope. I won't go into details, but he regarded images as pure data. So you can think about it. And I want to exemplify this in DNA when you think about DNA sequencing, because you use this tech for typically DNA sequencing. This is a part when you sequence a DNA. What you generate is images like this. Very interesting, no? But you just keep on producing these images and plug it into a computer, and then the computer starts to sequence everything, all of your genes, all the bases in your DNA, 3.5 billion bases. And you get it out. You can get all your genetic information. Your grandparents were from Greenland or whatever. So that's one typical application. Another application is when, as you saw in the brain image before, you actually want to see the image. You want to have localization. And you also want to see the genes, where they are located. So then this is kind of in between. It's both the image as an image and the data. So you also get genetic information. Sequencing, we'll focus a little bit more on sequencing here. Today, the challenge is to make it cheaper and make it faster. So it takes about 3 days to sequence you, and about $1,000 to do it. So be careful if it's cheaper. Then you know it's not sequencing. Okay, and I was in California like a month ago, and what's happening right now in California? That's actually So the question is why. Why would you want to make it cheaper with sequencing? Why would you want to do it faster? Here's an example of why you need it. So this is a company called Grail. They are co-founded by Google. And the Grail, the name is, they want to solve the Holy Grail. Basically, they want to take a blood sample from you, and then they want to sequence everything in that. And it turns out that if you have cancer, some of the DNA belongs to the tumor cells. Probably heard in the news like 2 weeks ago about mothers with babies. You can detect DNA from the blood, samples from the baby, from the fetus, and then you can, draw different conclusions. But in this case, they are looking at tumors instead. And in this case, they have to do really deep sequencing. They have to do it repeatedly over and over again. So it costs a lot of money. Basically, the CEO who used to work at Google, Jeff Hoover, he said that it costs like a car for each blood sample. So you can imagine how expensive it is. So you really want to make it faster and cheaper to be able to do this. And then you can use single technologies. Our little machine, model Tata, which is extremely fast. So you can you can do this digitalization process or sequencing process very fast. I won't go into detail, but it's like 1 trillion data points that we can generate, and about, I think we can go up to 1 billion data points per second, or pixel if you want to regard it like that. So it's way much faster than anything existing right now. And we also have this very sensitive imaging device, but I won't go into details about that. It has to do with price for sequencing. So to kind of round off after 10 minutes, that's that's good. So I wanted to, so right now you have the next generation sequencer or DNA sequencing, and the reason, I mean, some time ago when they started this human genome project where they want to sequence the whole, it costed like $1 billion, it took forever, so it wasn't really. But what happened was that they invented smarter imaging system or faster systems, of course chemistry too, and then they adapted computers. So they generated data and they let computers do the calculations, and then it opens up new opportunities for doing DNA sequencing extremely fast. That's what happened, as I see it. And this is just the start. This is the same concept will happen in a lot of different areas. The guy before, he was talking about proteomics. You can do similar stuff there. Just generate tons of data on proteomic data and feed it into computers and let them draw a conclusion of complex problems. I want to say one thing. I actually forgot about when it comes to Grail. So a step back. When they do sequencing, there is just a tiny, tiny fraction of the DNA that actually stems or comes from the tumor. So it's very difficult to just sequence a patient or a couple of patients and try to draw a conclusion about whether you've got early cancer or not, because that's the Grail. You want to identify cancer early. So you can do something about it. If it's late stage, then it's very difficult. So you want to do it in early stage, and that's what they aim for. And why Google is into this is because they need to be able to tell whether you've got cancer or not. You need to train an artificial, a computer, an AI kind of neural network, or how would you describe it? And to be able to do that, you need probably 100,000 patient samples, probably at least. And then you can imagine if the price is like a car for each sample, you need a lot of investment. I think they raised $900 million, this company. So you can imagine why they did it. I go back, I go forward. So I think this is just a start. And to round off this talk, I just want to mention a new project which is called the Cell Atlas. It's a lot of atlas nowadays, but it's kind of interesting. It started, I think it was Johan Strömqvist that took the initiative, to some extent at least, to identify every cell type in our body, basically everything. So that it's like the new human genome project, but now determine cells. And there's trillions of cells. It's horribly difficult to do this. It will take a lot of time if they don't use our systems, of course. So they are they're using different methods to determine these different cell types. You can't just look at the image. You also need either sequencing data or some kind of proteomic data to determine whether what type of cell it is. And I was surprised, and I'm going to end with this, because so I had a friend say, yeah, okay, so what's the reason? What will this result in? And I don't know what it will result in, just like I didn't know probably when NGS, DNA, the Human Genome Project started. So, but I talked with one of the professors, and he said that in the brain, there are like 200, 200 types of neurons, types. And and my idea before this was like, it's neurons. So if there are 200 different types or more neurons, who knows? Do we even know how the brain works? That's kind of these kind of questions that I get. So I'm really looking forward to see what's happening with this project. And by that, I will end my talk. Thanks.